Hello everyone, my name is Will and today I will be presenting my fMRI research proposal, the neural correlates of a political referendum, an fMRI study on in-groups versus out-groups. The human brain has developed specialized areas involved with social living, such as the amygdala, which is important for emotional processing, and the orbitofrontal cortex, which is involved in decision making. These areas are implicated in our responses towards and processing of in-groups and out-group members. In-group bias can be both implicit and explicit. We can categorize ourselves into groups based on things such as race, age, sex, and even political orientation. This bias can extend beyond this to cues and representations of a given group. This includes things like flags, colours, and t-shirts. Despite there being numerous studies examining the neural correlates of outgroups and in-groups, there is a lack of fMRI-centered research examining the neuroscience of cues and representations in the context of outgroups and in-groups. The 2023 Australian vote to Parliament referendum poses a unique opportunity to study these outgroup and in-group cues and how they're processed in the brain. Our study aims to fill the gap in the literature by examining individuals involved in the referendum. We hypothesize that exposure to vote supporting stimuli such as t-shirts, flags or signs will be correlated with a measurable increase in blood oxygen level dependent signals in both the amygdala and orbitofrontal cortex relative to baseline conditions containing vote neutral visual stimuli. As well as that exposure to opposing visual stimuli, for example, presentation of no related stimuli to a participant who voted yes, will result in less significant increases in bold signals in both the amygdala and orbitofrontal cortex relative to vote supporting stimuli. So for this study, we plan to recruit 50 participants, half of which will be no voters, the other half will be yes voters. Participants must be of voting age and will be screened for pre-existing psychiatric and neurological conditions. The study uses a block design and is comprised of 4 runs. Each run will contain 9 blocks. 3 blocks will have vote supporting stimuli, 3 will have vote opposing stimuli, and the last 3 will have vote neutral stimuli. Each of these blocks will have 6 trials. So here is an overview. So I've got 6 trials per block. So vote supporting, vote opposing, vote neutral, and so on. So here's an overview of the conditions and stimuli. So we have the vote supporting condition, which is yes vote related images for yes voters. And no vote related images for no voters. So that is basically flipped for the vote opposing condition, and then non-vote related images for neutral for both yes and no voters. So for each trial, the stimulus images will be shown on a screen. This will be presented. So for each trial, the stimulus images will be shown on a screen. These will be presented in grayscale and will have standardized image resolution and size. Here's an overview. So it's a fixation point here. And here's an example of a neutral stimuli, which is basically just like a grayscale shape. And we have either a negative or positive stimuli here, depending if you're a yes or no voter, which is just a t-shirt with the word yes on it. This is sandwiched between 2 1 second black screens, just so we can return to baseline before the next condition, before the next stimulus. So a repeated Latin square design was proposed for this study, although it may be simpler to utilize a within block randomization. This means the trials within each block will be presented randomly. This will control for learning effects and result in more generalizable findings. As for data acquisition, in this study we'll be using a 3T MRI scanner. T1 structural MRI will be acquired prior to any of the runs. This study will utilise a T2 star weighted images as this best shows the hemodynamic response to the stimuli. So here's an example of the block design and the recorded hemodynamic response in response to the given stimuli. And here's just an overview of the acquisition, the region of interest, amygdala and the orbitofrontal cortex. Got a repetition time of 2 seconds, an echo time of 30 milliseconds, and a flip angle of 90 degrees. So pre-processing data will be done using FSL software. Data will be corrected for head movement, non-brain voxels will be removed, and field maps will be acquired to address the inhomogeneities that result from surrounding tissue. Acquired images will be transformed to a template T1 weighted brain to allow for a comparison between participants. So here's an example here, raw fMRI data. This is the data after brain extraction, so those non-brain related voxels are removed, and then a correction of the image to a standard space. Here's an overview of the non-imaging demographic data. This will be collected during recruitment. A mixed ANOVA will be utilised to examine the between group differences and within group differences. The analysis will allow us to examine how factors such as age, gender, ethnicity and education affect brain activity in response to the presented stimuli. As for the imaging analysis, to identify how the brain responded to the given stimuli, the stimuli being vote supporting, vote opposing and vote neutral, a contrast of parameter estimate or COPE will be utilised. This will be used to compare neutral activity between the conditions. T1 weighted brain, T2 weighted brain, and this is just showing a bold signal in the amygdala on a T1. A random effects model will be used to examine variability between different groups. This will allow us to generalize the data to the population. To ensure reliability of results and to minimize errors, a family-wise error correction and thresholding will be applied. This will allow us to identify significant activations in key brain areas, specifically in our regions of interest, the orbitofrontal cortex and the amygdala. And just here's an overview of the COPEs. So it's just allowing us to contrast the parameter estimates, allows us to compare activity between conditions. As for budgeting, the total estimated cost of the study is roughly $8,800. This covers the cost of the MRI scans, the participant compensation and lab assistant pay. Participants will be compensated with a $50 voucher. And a lab assistant will be needed over a period of 2 days. So in conclusion, through examining voters from the Australian 2023 referendum, this study aims to examine how in-group and out-group associated cues are processed in the brain. It should be noted that this study could be altered to examine individuals supporting things like a sporting team, so it doesn't have to be it can be replicated in different situations. To achieve our objectives, we plan to utilise fMRI techniques. Through analysis of the acquired data, we will compare the bold responses to stimuli between individuals of the yes and no groups. Using the COPE analysis, we can identify variations between conditions. Using a random effects model, we can account for the variability between groups and generalise our findings to the wider population. By examining how cues that are related to relevant in-group and out-groups are processed in the brain, our study will address gaps in the literature. This has potential for to further develop our understandings of social dynamics and in-group bias, paving the way for future studies in this area.